Last week, you put together a molecular phylogeny reconstruction for a clade of different species that you selected to focus on as your little Bio202 project. It's not that many years ago that what you did in our 3-hour lab would have been the basis for a person's master's thesis. And yeah, now it's a lower division lab exercise. Okay, so you had some help. Someone else did the grunt work of collecting and sequencing the molecules. That is, getting the actual sequences that you compared. But actually now, this is also something we could totally do in our classes. In fact, when you take Bio 204, you'll be starting with tissue samples, and then you'll use PCR to prepare DNA for sequencing. At this point, we're still outsourcing the actual sequencing steps, but the costs of next generation sequencing tools are almost within reach for us. Maybe not something we can have on our campus yet, but I predict that within 10 years, in-house sequencing will be there as part of the Bio 204 curriculum and also, of course, our biotech curriculum. Now, what you did in your lab exercise was based on comparison of sequences that were in the range of several hundred to several thousands of bases in length. That's enough to do phylogeny reconstruction. And up until recently, we were in a place where using longer sequences might allow for somewhat greater confidence in the results, but with the older Sanger methods for sequencing DNA, the added expense was really not justified. If a sequence of 1140 bases gave us good results, we didn't have much incentive to spend the money for longer reads. With a new technology, NGS stands for Next Generation Sequencing. Long reads are not only possible, they're also relatively cheap. This has precipitated a next generation problem. We've gone from comparing and analyzing similarities from sequences of a few thousand bases, a lot of data, but manageable, to having ready access to the full genome sequences, hundreds of millions to billions of bases. To put this into perspective, 1,000 characters would be about 4 or 5 of Trump's tweets. That's like the sequence that you used for your phylogeny reconstruction. Now, 1 billion characters is 294 King James Bibles. That's like the sequence length you get from an NGS long read. How do you deal with this much information? Now, you can't just scale up the same kind of analysis that we did because the computing time required to do the same processing algorithms would be insane, beyond millennia, even with the fastest computers. It's a fascinating problem and one for a new breed of biologist who's versed and skilled in handling this kind of big data. Now here's another expansion or extrapolation from what we did last week. So from your phylogeny reconstruction, you put together a reasoned guess for the evolutionary history for a handful, 6 or 7 species. Now this could theoretically be extended to a much larger array of species, and unlike increasing sequence length by a millionfold, we could totally do this. This is something that existing computers could totally handle. Heck, we could even do all of life if we could find a sequence that were homologous for all living organisms. Something that both you and E. coli share because you both inherited it from your most recent shared ancestor from billions of years ago. But are there genes that you share with the bacterium? Well, it's funny you should ask. There are several, and these are basically the foundational evidence supporting a single origin for cellular life. Presumably, if modern life had descended from different points of origin, if life started twice, and one origin gave rise to bacteria, and a different origin gave rise to us, there would be no expectation of strong commonalities between bacterial cells and ours. In fact, there are dozens of similarities, most aspects of the genetic code, for instance. Now, the only reasonably parsimonious explanation for all of this similarity is that all of known life has a single common origin, and there must be a most recent common ancestor that connects all living organisms. Starting with this premise, you should realize that some molecular evolutionary biologists must by now have attempted a reconstruction for the phylogeny of all of life, using molecular sequences that are held in common by all living forms. And one example of such a sequence is that of ribosomal RNA. Ribosomes are needed by all cells to translate the language of genes into information that can be used to manufacture proteins. This is a basic functionality of all living cells. Ribosomes have many parts, an array of ribosomal proteins and ribosomal RNA. Now, one of the first attempts to reconstruct a universal family tree that connects you with E. coli was done by Pace in 1997, and it used the small subunit ribosomal RNA sequence. The layout of this tree is different from the ones we made last week. Rather than starting from a common ancestor on the left, here it's in the middle, where the three domains diverge. Eukarya on the lower left has a healthy number of species, and if you see the name Homo, that's you, right next to Coprinus, which is a kind of mushroom. Basically, animals are represented on this tree by, well, humans, and that's it. Realize that this was way back in 1997. It's just an early tree, a first step that focused more on relationships among the major domains of life. And at this scale, the differences between different forms of animal life, such as between you and a moose or between you and a cockroach, may not be all that salient. 11 years later, David Hillis puts his lab's tree, also based on ribosomal RNA sequences, out there to evoke shock and awe. This tree has over 3,000 species, including several animals. You can think of this as a group photo, and you know you're somewhere in the picture, so the first thing you want to do, of course, is to find yourself. Zoom in right where it tells you to look, and there you are, sharing a branch of the tree with Mus musculus, Rattus norvegicus, and Oryctolagus cuniculus. Right now, Mus musculus is the house mouse. Rattus is the Norway rat, which is also the same as the lab rat and the New York City subway pizza rat, and all the rats that eat my tomatoes from the garden before they can reach being vine ripe. Oryctolagus is the standard European bunny rabbit. Now, the four of you, the four species on this little branch of this huge tree, are the only mammals represented in the Hillis tree. Yes, if you're looking for patterns across all forms of life, representing lineages that diverged billions of years ago, differences among mammals, most of whom diverged a paltry 60 million years or so ago, are pretty trivial. If you want to focus on mammals, you'd need to narrow your scope, and you might want to use a molecular sequence that's well suited for the comparisons that you need to make. In other words, do something like the activity that we did last week. Okay, here's a question for you. Looking at the branching pattern among these four mammals, as shown in the Hillis tree, which species is most closely related to humans? Go ahead and look at this carefully and tell me which is our closest relative, the mouse, the rat, or the rabbit. I hope you said none of the above, or all of the above. They're all equally related to us because they all share a most recent common ancestor, this guy right here. That's not in the human lineage. For the clade of rats, mice, and rabbits, we are the outgroup. Now, of these three non-human mammals, which two are most closely related? Yeah, easy. Mouse and rat, which makes sense because they are both rodents, while the rabbit is in the closely related non-rodent taxon the Lagomorpha. Together, rodents and lagomorphs are their own clade, the Glirras, which is a sister clade to the primates, which is our group, of course. Glirs and primates form the core of the Euarchontoglirs, which is one of the four major clades within the placental mammals. Now let's look outside of mammals. Who are our closest non-mammalian relatives represented on this gigantic tree of Hylas? Well, the next most recent node on the tree connects us with this other clade running from Pseudomys scripta, which is the red-eared slider turtle, all the way to Alligator mississippiensis, the American alligator. One cool thing to note here is that the alligator's sister taxa is this pair of species, Turdus migratorius and Gallus gallus. Now, Gallus might be recognizable to anyone who speaks Spanish or Italian. El gallo or il gallo means rooster, and so Gallus gallus is the species for the chicken. Turdus migratorius is kind of a curious name. Literally, migrating turd, but this is the species of the American robin. So robins and chickens, both birds, are together as their own group. Makes sense. But according to this, their sister taxon is the alligator, and the alligator sister taxon is the birds. That is closer to the alligator than the turtle. Now, what are these other three species? Sphenodon punctatus, Heterodon platyrhinos, and Sceloporus undulatus. Well, Heterodon and Sceloporus are the two most closely related to each other, and they are the eastern hognose snake. That's Heterodon. It's a crazy animal that is on one hand a mimic of dangerous snakes. It's non-venomous, but it resembles a rattlesnake in its coloration pattern. And if that defense doesn't work and the predator decides to attack it, it goes into a bizarre death-feigning behavior, writhing upside down in a sham epileptic seizure while gagging up mucus in a way that totally discourages predators. I'm guessing it just grosses them out. Anyways, you have the snake and also the fence lizard. Sceloporus undulatus is the eastern version of the western fence lizard, Sceloporus occidentalis, that is common in our area. I see them both at home and on campus all the time. Okay, so that's Heterodon and Sceloporus. Sphenodon is this totally weird animal that looks more or less like a regular iguana, but on closer inspection, they're actually not regular lizards at all. In fact, they are the sister taxon to the squamates, which is the clade including all lizards and snakes. A regular iguana, which is a lizard, is more closely related to a king cobra than it is to the sphenodon thing, which is also called a tuatara. So from this, it looks like the birds are nested. Get it? Nested? Within this larger clade that includes things that we normally identify as reptiles. The turtle, the lizards, and the snakes, and their sister, the sphenodon, and the alligator. And this might look weird to you right now, but after Bio 202, it won't be weird at all, because you'll know that the birds are just modern dinosaurs. And we're just fine with dinosaurs being evolutionarily close to alligators, aren't we? Anyways, this really has little to do with today's lab, but it's more like a follow-up to our exercise from last week. But now, if you look at the whole structure of this tree, you can see that it attempts to do the same thing that the pace tree from 1997 did. Or this tree by Carl Woese that we should have already seen in lecture by this point. The pattern of phylogeny beginning from a single starting point, a single origin of cellular life. Today's lab, the microbes lab, normally is constrained by the limits of what can be done practically by students in a teaching laboratory of modest means. This semester, we don't have the opportunity to be practical, and this has some downsides. For example, you won't get valuable time using the microscopes and preparing slides for viewing. You won't get the chance to learn how to recognize Pseudomonas aeruginosa by its smell. On the other hand, we don't have the constraints that we normally do, and so we can go nuts a little and address taxonomic groups that we talk about in lecture but could never provide in a real-time lab experience because the cells are impossible to get, require exotic conditions, or in some cases are just too dangerous to handle. So, I'm thinking that by now you should be familiar with the basic layout of the prokaryotic side of the tree of life. We start from that universal common ancestor, and on one side, the archaea side, we have the euryarchaeota as the sister to the clade consisting of the lokiarchaeotes and the tacharchaeans. The euryarchaeotes have two well-known forms. One that is largely a specialist on extremely salty environments, the halophiles, and the other one is notable for being able to produce methane gas. Those are the methanogens. On the other side, the lokiarchaeotes are placed basally with respect to the main tac supergroup. Remember, the lokiarchaeotes are presently considered to be in the same archaean lineage that gave rise to the cell that became the first eukaryote. And by that token, these are the prokaryotic cells most closely related to you and me, or at least the main part of our cells. Other parts of eukaryotic cells, like the mitochondria, derive from other different endosymbiotic microbes, all of which are bacterial or, in some cases, eukaryotic. Now, in the TAC supergroup, the only one we care about, which is also the most well-studied, are the thermoacidophilic Crenarchaeota, the C in TAC. And these are most famous for the requirement of superhot and or superacidic habitats. Think of boiling battery acid as preferred conditions of temperature and pH. But don't think that you'll actually find them in a lead acid car battery. Metals like lead are pretty toxic to most kinds of life. And so, as you're thinking about what we're going to be doing in our virtual lab in support of the archaea, I want you to totally free yourself from the constraints of what we could do in an actual college laboratory. Normally, we might try to dazzle you with pictures or maybe prepared microscope slides of the organisms that we're learning about. These archaeal things. Okay. I'm just kidding about this being dazzling. Honestly, prokaryotic cells don't make for such visually engaging instructional content. But just to be on the safe side, I went ahead and posted a few still images on the Microbes Lab discussion forum. You don't need to do anything with them. As I said, they're there mostly just for fun. You can add a comment to identify which organism belongs in which archaeal clade. What I want you to do for this activity is to plan a virtual Archaean collection safari in which you and your team can go anywhere in the world. Limit yourself to the Earth, please, to collect a sample of live cells from nature for each of the following archaeal groups: extreme halophiles, methanogens, lokiarchaeotes, Crenarchaeota. You'll need to do a little Googling. Team up with one or two others if you like, group sizes of 1, 2, or 3 again, and put together a brief summary that includes precisely where you would go. And remember that you need to go to a natural source, no samples from laboratories or other human curated collections. Which species would you expect to find at that location? Normally, I would ask you to also include an image, but microbial pics are pretty boring, and so we'll keep it short and easy. Just exact location and what species you're getting there. This shouldn't take much effort. Your demographic tends to be really good at Googling. But now it's time for us to move into a virtual bacterial collection safari in which you and your team will do the same for each of our 5 featured bacterial clades: spirochetes, Chlamydia, cyanobacteria, Firmicutes, proteobacteria. This time, however, I actually do have some video clips of live bacterial cells in the discussion board for you to take note of. There's a time-lapse video of Chlamydia trachomatis destroying its host cells. Now this shows a membrane-bound vacuole within which the chlamydial cells are doing their thing as they reach the point of bursting the host cell apart. In lecture, we'll be thinking about what makes chlamydia a bacterium and not a virus. An important distinction because both of them attack a host cell and use it to make more of themselves, killing the host cell along the way. Another thing we'll talk about in the lecture is the bacterial flagellum. And we've done this. We've already done this in many different contexts. There was the War on Science video with Ken Miller teaching us about how a bacterial flagellum is in fact reducible and therefore not a good example of what the ID creationists need it to be. It's also no small deal that the bacterial flagellum is based on a true rotary mechanism, one of the few wheels that exist in the biological world. This is another cool thing about them. There's also the way that bacterial flagella are different things altogether from what we identify as flagella on eukaryotic cells. This is going to be another important point. So this microbes laboratory is usually a great opportunity for you to get a little glimpse. Perhaps not a very satisfying glimpse because you can't actually see flagella on live bacterial cells with light microscopy. They are way too thin, something like 12 nanometers in diameter, as compared with the minimum resolving power of even the best light microscopes, which is in the range of around 200 nanometers. Now, despite the fact that you can't actually see the flagella, you can tell they're there or not by the way the cells move. Non-motile bacteria that are either non-flagellated or maybe flagellated but dead or inert or lazy or something, they will still be moving when you look at them in a wet mount, suspended in water, but their movement will be Brownian, that is, randomly shifting or jiggling up, down, right, left. This is the result of the cells being continually bombarded by molecules of water. Bacterial cells are small enough to be affected by the kinetic energy of individual water molecules. By the way, this explanation for the random jiggling, first described by Brown in the early 1800s, this was reasoned out by none other than Albert Einstein in 1905. In addition to the Brownian movement, the bacterial cells in a wet mount preparation, again, that's when you suspend the cells in water and then sandwich this thin layer of water and cells between a glass microscope slide on the bottom and a cover glass. These particles will usually be subject to currents of moving water in that thin layer. It's weird but true that the water between the slide and the cover glass is not still. Basically, you have evaporation causing a loss of water at the edges of the layer, and so water tends to move from the middle part of the sandwich to replace the water that's lost at the edges. So at the same time these non-flagellated cells are jiggling randomly, shifting slightly in all directions, they might also be floating down current as the water moves across the field of view in your microscope. One of the challenges that microbiology students have to learn is to distinguish true motility from this combination of random jiggling and moving downstream. It's really not that hard. If all of the cells are going in the same direction, that's your downstream movement. You need to narrow your focus on a group of cells as they float downstream and determine whether they're just randomly jiggling or doing something else, something that would require flagella. Now, flagella, when they're there, can be in one of two different arrangements on the cell. Both are pretty common. The flagella can be all at one end of the cell. We call this polar flagella because of the way it sets up a front end and a rear end for the cell, its axis of polarity. Cells with polar flagella typically run very fast in one direction, occasionally stopping and spinning around. Okay? When you see this type of motility, you can usually infer that the invisible flagella that you don't see are all on one end of the cell. The other arrangement for these invisible flagella that you don't see is all around the cell. This is called peritrichous flagella. Bacteria with peritrichous flagella classically do more of a tumbling type of motility, like they're somersaulting around. Now, I'll admit that this is a little bit of a myth because there are quite a few peritrichous bacteria that swim perfectly well in straight lines. But in a teaching lab, I'm not supposed to complicate things like this. So in the discussion board video clips, the peritrichous cells will just be doing the tumbling thing. Now, in addition to these standard kinds of motility, I will also put on some videos of some less typical but still awesome kinds of motility. There's periplasmic flagella of the spirochetes, and also gliding motility, true motility by cells that have no flagella at all. It's pretty characteristic and nothing like any of the flagella-based mechanisms of motility. So besides maybe posting some identifications of what it is that we're looking at in the discussion board, there's nothing for you to turn in for this part of the laboratory where you're looking at the videos that I've posted. But know that for the test, you may be asked to view a video and answer a question about it, such as determine from what you were seeing the presence absence of flagella, and if present, what is the arrangement of flagella on the cells. Okay? Now, the last thing we do in a normal lab activity in a normal semester is the catalase test. This is actually a pretty important anchor for that part of the lecture when we talk about the dramatic shift in atmospheric chemistry at the Archean Proterozoic break 2.5 billion years ago. Before that time, no O2. After that time, O2. And this was a game changer for life. In particular, we stress that O2 is a very harmful and toxic substance for the kinds of cells that had been around for the past billion plus years, from the origin of life until that great oxygenation. In order for living cells like those of our ancestors to persist in the presence of oxygen, we had to evolve defenses. And one of the defenses that we're most familiar with is the enzyme catalase. Catalase, the enzyme, catalyzes the conversion of peroxides like hydrogen peroxide, H2O2, into something less dangerous. Peroxides form spontaneously in the presence of O2. Even hydrogen peroxide, H2O2, forms at very low levels when O2 and water combine. The problem with peroxides is that they break into free radicals. H2O2 splitting symmetrically gives rise to two hydroxide free radicals, HO dot, which are one electron shy of becoming hydroxide ion, HO minus. This means that HO dot is super electron hungry and very reactive. It will steal an electron from anything that it touches. We call this oxidation. And when it happens to one of your proteins, it causes damage. And if the hydroxide free radical oxidizes DNA, the damage may take the form of mutation. Basically, O2 is dangerous to unprotected cells because of all the pathways leading from it to the formation of these dangerous things like free radicals that destroy the cell's essential functions by destroying proteins and nucleic acids. At present levels of oxygen, 21% of our atmosphere is oxygen. Unprotected bacterial cells die immediately upon contact with ambient air. Your cells don't die because they're loaded with catalase. Even if a peroxide were to form inside of your cells, it would never have the chance to form a free radical because it would be immediately attacked by catalase and instantly converted into the relatively harmless forms of water and O2. Now, you've experienced this if you've ever had a cut or open wound that you've disinfected with a 3% hydrogen peroxide. That's a standard brown bottle from the drugstore concentration. You pour the H2O2 solution on, bubbles pour out from the cut, right? Those bubbles are O2. Now, you might have thought, maybe somebody told you, that the bubbles you see are bacteria in the wound dying. Maybe it's their screams as they're being massacred by the peroxide. No, that's not the case at all. The bubbles are from the reaction between the peroxide and the catalase that was there in your cells. Instead of the bacteria screaming, it's more like your cells are saying, "Bring on that oxygen poison. I've got this covered. Hey, unprotected bacterial cells, how you doing over there?" Right? The unprotected cells would, of course, be toast. But many bacterial cells are also protected by catalase. Now, we test for a bacterium's production of catalase in the most direct and obvious way conceivable. Open the petri dish and pour on 3% H2O2. If the cells bubble vigorously, does that mean the bacteria are dying? If the cells don't bubble at all, does this mean the bacteria have catalase or lack catalase? If the cells bubble, but only just a little bit, why would this result be consistent with a cell that's able to tolerate O2 but usually lives in an anaerobic location, like your gut? These last three questions are the ones you need to answer on your worksheet. That is, after having completed your Safari-based tasks from the earlier part of this lab.